Hi everyone, you are welcome to Biochemistry Basics here. My name is Motika Bertrand. So today we are going to be looking at proteins. But before we dive into proteins, let's look at what is known as a proteid. So a proteid is a polymer that is formed when two or more amino acids they condense together, leading to the formation of peptide bonds. Now these proteids are classified into two broad categories. We have the peptides and we have the proteins. What's the difference between a protein and a peptide? A peptide is a polymer that is formed when two or more amino acids, usually between 2 to 50 amino acids, they condense together, leading to the formation of peptide bonds. While a protein is also a polymer that is formed when more than 50 amino acids they condense together, leading to the formation of these peptide bonds. So they only differ at the level of the number of amino acids that are condensing to form these peptide bonds. While for peptides, we have from 2 to 50, then for proteins, we have more than 50. Now, the basic building blocks for these proteids are amino acids. So amino acids, they are the building blocks for these proteids. And amino acids, they have this general formula. So they have the amino group, they have the carboxylic group, they have this hydrogen, and they only differ now at the level of this R group here. And naturally, there are 20 naturally occurring amino acids, as we are going to see. And for us to be able to form a peptide bond, we must have two or more amino acids that are condensing together. And this one here is a simple illustration of the formation of a peptide bond. So we have the alpha carboxylic group of one amino acid reacting with the amino group of the neighboring amino acid with the elimination of water to form this peptide bond. Now, there are different levels of organization of proteins. We have the primary, we have the secondary, we have the tertiary, and we have the quaternary, as we are going to see. For the primary, it simply refers to a sequence of amino acids arranged together, sequence of amino acids, or the linear arrangement of amino acids in the sequence. For instance, you can have alanine, valine, serine, leucine, arginine, and so on and so forth arranged together. But now, this primary structure determines the structure and the function of the particular protein. Because a change in only a single amino acid, for instance, if I replace this valine here with, let me say, isoleucine, is going to completely change the shape of this particular protein, and it will obviously change the structure, the function of that particular protein. And the kind of bonds that are stabilizing this primary structure here, they are irreversible covalent bonds. You have to take note of that. Then for the secondary structure, it simply describes the local and the regular folding of the polypeptide chain, or it describes the localized shape of that particular protein to either give you the alpha helix or the beta plated sheets. For the alpha helix, we are going to obtain a protein in this kind of form, where we have an amino acid in one part of the chain reacting with another amino acid in the other part of the chain. And this one here is simply hydrogen bonds. So the kind of bonds that stabilizes secondary structure, they are hydrogen bonds. As you can see, the oxygen of this carboxylic group of one amino acid in this end here reacting with the hydrogen of this N-terminal of the next amino acid in the same chain. So the kind of bonds that are this kind of secondary structures, they are known as hydrogen bonds. And for each residue, each of these 10 here is made up of 3.6 amino acid residues for each 10. Now for the beta plated sheets, we also have something of this kind of structure where we have the carboxylic group of one amino acid reacting with another hydrogen in the N-terminal of the neighboring amino acid to give you this kind of hydrogen bonds. So the hydrogen bond that are stabilizing the beta helix, the beta plated sheets, is the same kind of hydrogen bond that are stabilizing the alpha helix. And then for the tertiary structure of proteins, which is the three-dimensional structure of the protein, it is the stage at which the protein is functional. And here, you can have a simple demonstration like this other one here, where we have one part of the protein is having the alpha helix, while the other one is having the beta plated sheet. And this 3D structure of the protein is the one in which the protein is functional. And the folding pattern of this particular protein here is more intense compared to the secondary structure. And when we have more than one tertiary structure that are reacting, that is, we have one of these tertiary structure, more than one that are reacting together, it gives us what is known as the quaternary structure, which are several of these polypeptide chains that are interacting. And an example of that is hemoglobin, which have 4 different polypeptide chains, 2 alpha helices and 2 beta helices that are interacting to give the functional protein, which is hemoglobin. We also have insulin that is made up of 2 tertiary structures or 2 polypeptide chains that gives the structure of insulin. So the state in which a protein is functional is only in the tertiary stage. So I hope this video was helpful, and if you like this video, please subscribe, share, and make sure you comment. Thank you, and see you in our next video.